In this video, we will discuss about transduced allogenic NK cells results in the NSG mice with MHC1 and MHC2 double knockdown. So basically, they are checking the efficiency of their transduced allogenic mice that has the lower expression of human leukocyte antigens ABC and high expressions of human leukocytes antigens of E type. That is, they have this guidanase 13 that is used to now suppress HLA-ABC, and this SC that is used to express HLA-E antigen. And in the in vitro system, this model gives the best results, so they wanted to check its efficiencies in the in vivo system. So for that, they use this NSG mice that has the low immunogenic response, that is, it lacks the B cells and the NK cells, so that the immune system of the mice does not attack the immune system of the humans that will be injected in this. And to prevent the interactions of immune system of the humans to the immune system of the mice, they use this MHC1 and MHC2 knockdown, because the human T cells, that is CD8 plus T cells, recognize using MHC1, and CD4 plus T cells recognize using MHC2 the immune systems or immune cells of the mice. Once they recognize, they will generate the immune response. So in order to suppress that and to have a long-term studies of these mice, they use MHC1 and MHC2. And this is the overview of the plan, that they use NSC mice with MHC1 and MHC2 knockdown and did this irradiations of this 100 CGY unit just 2 days before the injection of this mixture of the T cells and the NK cells, and then do the measurement just before the injections at this 724 and 21 749 21 days using the flow cytometer. And at the same time, they also introduce this IL-2 every 3 days, the recombinant human IL-2 every 3 days to keep the NK cells active. And they use the combinations that in the one combination they use donor 1 NK cells and then use donor 2 transduced NK cells. So this will act as a allogenic NK cells, the donor 2 untransduced allogenic NK cells. And what they're able to see, and at the same time they also mix these cells with the T cells of the both donor 1 and donor 2. And in the first case, that they are able to see that since the T cells of the donor 1, in this case they use the T cells of the T cells and the NK cells, yeah, both of the donor 1 and donor 2. And so in the first case, what they are able to see is that the percentage of NK cells. So let me again explain. So what's the things that they have injected in this case? This is panel D and E. They use donor 1 NK cells, donor 2 untransduced NK cells, and donor 2 untransduced, donor 2 transduced NK cells, and donor 2 untransduced NK cells, and the T cells of this donor 1. And what they are able to see is that NK cells in the NK cells just before the day 1, they are able to see a suppressions of the donor 2 untransduced NK cells, indicating that the T cells is suppressing or killing these untransduced NK cells, but they are not affecting the transduced NK cells. In fact, the transduced donor 2 NK cells is increasing. And this thing they have checked it in the blood, and when they check it in the tissue in the forms of, they check these NK cells in the tissues, but they are able to see a consistently maintained donor 2 untransduced NK cells that can be seen here, with expressions of around 80% in almost all the tissues that they have checked. It is lung, liver, spleen, bone marrow. And when they did the reverse thing, that is when they use donor 2 NK cells and the donor 2 T cells and the donor 1 untransduced NK cells and donor 1 transduced NK cells, they are able to see the similar results. That is the suppressions of the allogenic donor 1 untransduced NK cells, but the allogenic donor 1 transduced NK cells increases in the blood and maintain the same level, almost the same level in the different tissues. And thereafter they checked the T cells efficiencies, so in the form of T cells, and these results are shown only for the first case. in this main figure, that is T cells. So before transductions the T cells are around 25%, but they are increased before day 7, and they are maintained at almost at the same level for day 14 and day 21. And these things, the results are for the blood, and in the case of the tissues in the form of leukocytes, the human T cells, that is CD45 T cells, they maintain the same levels around 50% in almost all the tissues that they have observed. When they check the activated T cells, they are able to see that the T cells before transduction activations are low, but just before day 7 the activations are high. It could be because they have recognized the untransduced allogenic NK cells, and they are working to clear off those untransduced NK cells. And once they are cleared out, then this activated T cells again goes down. And when they check this activation of T cells in the different tissues, they are able to see almost similar amount of T cells, because the almost similar amount of activated T cells, that is around 15%, because the one of the reasons could be that untransduced NK cells might not have reached to that site, and even before that they are being cleared off by the this activated T cells, CD45 plus T cells. So this is what they have shown, that this indicates that the transduced or the optimized transduced allogenic NK cells can evade the host immune cell response and perform better compared to the untransduced allogenic NK cells. So this is what they have proved from the in vivo studies. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. One more thing before we end the video is that this figure is the continuations of the previous figure that we have discussed. Thank you for your time.